think we'll get going here. Let people finish getting their food. I'm filling in for Dr. Ram here, who's on vacation this week, and it's my pleasure to introduce Raphael Bernier, who is an assistant professor here at the University of Washington, I think located primarily at the CHDD, which resides behind the University of Medical Center's primary campus here. And he studies a number of things, but he's a PI on something called the Simmons Simplex Collection, which is a multi-site study of the genetics and autism, specifically looking at some neurophysiologic consequences of having genes and autism. He got his training here at the University of Washington and then went on to get clinical training at UCLA and has also been further east to Wisconsin and Tufts. So it's my pleasure.
Great, thank you. Excellent. Thank you. Welcome, everyone. Thanks for having me today. It's really my pleasure to talk about essentially my odyssey in trying to make sense of the variability we see in autism. So quickly just start by saying I have nothing to disclose. My funding comes from NIH, Autism Speaks, or Simon's Foundation, or the sale of my book. I've got no other conflicts of interest there. So making sense of the heterogeneity we see in autism. So one of the common phrases we hear is, you've met one child with autism and you've met one child with autism. And it's kind of a common phrase because there's so much variability there in terms of presentation. And that's not only in terms of presentation, but there's variability in terms of course and outcome. So for some kids with autism, impairments might continue on through life in a significantly impactful, impairing way, whereas other kids seem to be significantly impaired early on, but tend to sort of show some moderate improvement over time, while still showing some of the quirkiness we still see early on, but moving on. Similarly, we see a lot of variability in terms of response to treatment. Some kids with autism seem to respond very quickly, very rapidly to treatment, do very well, end up in a regular education classroom, whereas other kids with autism, we might intervene and those impairments persist and there doesn't seem to be any response to treatment. So trying to make sense of some of this heterogeneity clearly plays an important role in helping us figure out how we're best going to treat individuals with autism. So I'll start today just by talking briefly about the current sort of state of our understanding of behaviorally defined subtyping in autism, trying to parse out that variability. And then really going to talk about sort of my experience over the past 15, 20 years to try to make sense of the subtypes in autism from a more genetic first approach. Instead of thinking about it just from behavior, but actually look at genetically defined subtypes. And I'll talk a little about some of the research I'm doing now to help sort of further that process. So currently, the way we define diagnosed autism is simply based on the presence of impairments in social communication or the presence of restricted or repetitive intrusive behaviors. It's behaviorally defined solely. And for a long time, there's been attempts to parse this out into subtypes. There's, back in the 70s and 80s, 3 subtypes were proposed by Wing and Gould. They proposed that kids with autism could be sort of divided into subgroups of aloof kids who really just had zero interest in social motivation, zero interest in interacting with others. Other kids who seemed in some way interested in engaging in the social world. They termed them as active but odd, so they engaged with others in an atypical way. And a third approach that the Wing and Gould folks proposed was that there was a third group of kids who were just significantly withdrawn from the world. It wasn't necessarily that they had zero social motivation like the aloof group, but they were just more withdrawn. So that behaviorally defined subtyping process never really took hold, and the DSM-4 categories took hold instead, and that's been true for several years now. Those 3 subtypes that are behaviorally defined would be autistic disorder, Asperger's syndrome, and PDD-NOS. This will all be changing in a couple weeks, but this is where we've been for the past little bit. Now, what we've found since this publication of DSM-4 is that these 3 defined subtypes, well, they don't seem to match on to etiology in any way. They also don't seem to guide our choice in treatment or intervention. And thirdly, even the best diagnosticians in the country can't seem to actually figure out which kiddo with autism falls in these 3 different subcategories, really suggesting that these 3 subcategories aren't probably the best way to move forward. As such, the DSM-5 will actually be collapsing these three subgroups into one overall umbrella term, autism spectrum disorders. Which might make you think, gosh, aren't we going backwards away from subtyping into a more universal terminology? And I would say yes at this point, but this allows us actually to define more appropriate subtypes than we currently have. Let me give you a little example about some of the challenges with this behaviorally defined subtype approach. And it really comes from some of the work we were doing as part of the Simon Simplex collection. So this is a study that was started about 6 years ago, funded by a private foundation, and it was a study designed to bring together the top 12 diagnostic academic centers for autism in North America to work together to identify autism, genetic contributors to autism. And really, because we hadn't been doing that before that. Really, we were doing great work at UW, and Yale was doing great work, and UCLA was doing great work, but we weren't really working together, and we really needed to have these sample sizes large enough to really make sense of this complicated puzzle. So this private foundation, which happened to have enough money to provide the infrastructure and the enthusiasm for everyone to work together, allowed us to have this collaboration. And working together really meant working very closely together. So we collected every single measure across all sites in exactly the same way, and we had very strict cross-site reliability. We videotaped all our sessions. We sent out the videotapes that we collected here at UW to the folks all over the rest of the country, and we got their tapes, and we double-coded everything, and we made sure that we were doing everything exactly the same way. We had strict guidelines, strict reliability measures for our diagnostic tools. We used the gold standard diagnostic instruments of the Autism Diagnostic Interview and Autism Diagnostic Observation Schedule. We made sure that we were doing everything exactly the same way. Now, there's been a long history in genetic studies for us to use these gold standard diagnostic tools, but then also to include clinician diagnosis using the DSM-4 criteria. It would make sense. We're all expert diagnosticians. We are experts in autism at these 12 different centers. We should be able to use the DSM appropriately. So we didn't do anything specific around the DSM, do any specific reliability around DSM, because why bother? We were experts. We know this. DSM we thought was good enough. So when we looked at the data across all the sites, and what this slide shows here is just a handful of the hundreds of measures that we collected. On the x-axis, we have data points from each site. On the y-axis is performance on a given measure. So here, for example, we've got performance on diagnostic tool looking at social communication ability, social affect, looking at the social domain, repetitive behaviors, severity. And really, the take-home is that the kids that we were seeing were all pretty similar. So there's no one site that had kids that were more impaired socially, or no one site that had kids who were less verbal, no one site that had kids who were more hyperactive or more irritable, or any of these traits. Across all the different traits we looked at, here's just a handful more different traits we looked at, no one site was standing out. Across the sites, we were seeing all the same kids. So when we looked at our diagnostic breakdown of our different three subtypes, one might anticipate that we're seeing the same kids. We're all experts at diagnosis. We use the DSM-4 criteria. One might expect we'd see sort of similar rates of diagnosis at the subtype level. And clearly that was not the case. There were some sites, Site G, for example, didn't diagnose any of their kids with anything other than autistic disorder. They didn't see any kids with Asperger's syndrome or PDD-NOS. Other sites, almost 50%, were making a diagnosis in the PDD-NOS subtype. So this was kind of a sobering finding for us. We published this in Archives of General Psychiatry just last year. Kind of a sobering finding in that, gosh, we're supposed to be the best at doing this, and we clearly can't get it right. Anyway, this provides further support for some of the changes that are coming out really at the end of the month here in DSM-5. So, behaviorally defined subtypes, we're not there yet. I haven't been able to identify that yet. So really what I've been focusing on since I got involved in the autism world back in the 90s is thinking about how we can parse out this heterogeneity by identifying genetic subtypes, by understanding better the genetic contributions to autism. So my story starts back in the 90s when I took a gig with Susan Folstein, psychiatrist. She developed the mini mental status exam and did the first twin study of autism. And I joined on her team as a diagnostician and phenotyper. And what we did was we were conducting a linkage study of autism, NIH-funded study to look at multiplex families with autism, see if we could identify potential regions in the genome where there might be some candidate genes that we could explore. So as part of our study, I drove all over New England, all over the East Coast, working with multiplex families with more than one child diagnosed with autism. I would draw their blood, I would do a full diagnostic battery, and then we would actually pool our sample together and actually see if we can identify, are there any hot spots, any regions of the genome that might point to potential candidate genes. So we published our findings in 1999, and on this sort of old paper, it seems like 1999 wasn't that long ago, but even just looking at the images, it just seems like it's olden times. At any rate, we identified a couple of different regions on a couple of different chromosomes that were potential hotspots. There might be a gene or candidate gene there for autism. Now, subsequent to this publication, there's been a number of the folks that have published their linkage studies, and well, the reality is there was very little overlap with any of the work that we'd done or any of the other studies, really highlighting that not only are we seeing this phenotypic variability, but we're seeing a lot of genetic variability, right? A lot of different genetic contributors. So if you have a lot of different genetic contributors, there's not going to be one region or one candidate gene that's going to stand out that would give us a strong enough signal in a particular region, which suggests that perhaps if we're going to do a linkage type study analysis, we really need to have a lot bigger numbers, or we might want to think about a different approach to thinking about identifying candidate genes or risk loci. So while we were doing this multiplex study, though, working with Susan Folstein, and she says, Rafe, so I know this is great. We're doing this multiplex study. You're driving all over the place. We're collecting all this data. I think it's great, but I think we might really want to focus on also a different population of families with autism. We're focusing on multiplex families, but really that's really kind of a minority of all the cases of autism. The vast majority of families with autism, there's only one child that's been diagnosed. We don't see a lot of kids diagnosed with autism. She said, I think this might be the key. So can you go ahead and on top of what you're doing now, just go ahead and find all the simplex families, those families where there's one kid with autism, and go ahead and collect their blood and interview and do all your diagnostic workup that you do. And I said, okay, great. I'll do that. So went ahead and did that. And then I got married, and then I joined the Peace Corps, and so I bowed out of the scientific world for a couple of years and kind of left that data set just sort of hanging there while I was basking in the happy isles of Oceania. But work was continuing without me. And during that time, while I was in the Peace Corps, Jonathan Sabat and Mike Wigler, who are geneticists at Cold Spring Harbor, they were thinking, you know, folks have been doing this multiplex analyses for a while now. It's not really getting us very far. What about simplex families? Is there maybe a different type of transmission or type of genetic contributor in these different family types? Might that help us understand what the genetic contributors are to autism? So what they decided to do is they were interested in looking at copy number variation. So small structural rearrangements of the chromosome. And what they were interested in doing was looking at if individuals from simplex families with autism versus children from multiplex families with autism, might there be a different rate of these copy number variations, these tiny structural changes in the chromosome, deletions or duplications. And what they found was they looked at the typical population. They found that of the sized type copy number variation they were looking at, about 1% of all folks in the general population seem to have these copy number variations, these deletions, duplications. They looked at the multiplex samples. They gathered up a lot of different samples from a lot of different studies and looked at these multiplex families, and they found a rate of about 3% of these copy number variations and these duplications and deletions scattered throughout the genome. And then looked at the simplex sample that I'd collected back all over New England, and they found 10% of that sample seemed to have these copy number variations, these de novo or newly arising spontaneous deletions, duplications that parents weren't, that they didn't find in the parents. And it kind of changed the field in a way because it started us actually thinking a little differently about how to explore the genetics of autism. So Mike Wigler, this sort of geneticist at Cold Spring Harbor, he had a good buddy named James Simons, who was a wealthy billionaire who made a lot of money in hedge fund work and who had a daughter with autism. And Mike and Jim were chatting, and Mike said, I think this might be kind of the way to move forward. And Jim said, well, I don't really know anything about autism, but I do have a boatload of money, so I can help sort of make this work. So they put together the Simon Simplex collection, and were able to start working together again to identify. Primarily, we're aiming at identifying these copy number variations that we might find in these simplex families, with an eye towards a whole host of other things we could do with this sample as well. So we started the Simon Simplex collection in 2007. There were 12 sites. And as I mentioned, we were doing everything exactly the same way. We originally proposed to work with 2,000 families over the course of 3 years, but there was so much excitement in the field for this project, we ended up expanding to 3,000 families. We worked with 3,000 families with autism, collecting blood, collecting a massive amount of phenotypic data in just a short 4 years. It was kind of a monumental undertaking, and I think it shaved off those 4 years of my life. I think it's probably worse than cigarette smoking. That's my guess in terms of the stress, but that's okay. At any rate, so we actually just recently published the work that we've done here at UW, looking at our screening of these copy number variations, these deletions, duplications in this sample. And this is a very busy slide, which it's just one of the figures from our paper. This is work that I did with a great collaborator, Evan Eichler, here at the University of Washington Genome Sciences. And what we did for this paper is we just simply wanted to screen our sample of individuals from the Simon Simplex collection for the presence of these deletions and duplications. We also then compared it, and so that's highlighted in blue here on this very busy slide. The x-axis shows the particular copy number variation that we identified, as well as some of the genes of interest, and essentially the percentage, the number of times that we observed that mutation. That's in blue. In red, we also had another cohort of individuals with developmental delay without autism, as well as a cohort of controls. And so we screened through the whole sample to, again, identify these tiny structural chromosomal rearrangements. We found a number of recurrent events. The locus 16p11.2 seemed to be the most common finding for us. That is either duplication or deletion at chromosome 16p11.2. Other regions that were highly recurrent are events at chromosome 1q21, as well as events at 15q13. So that was just kind of our first stab. Let's just see if we can identify some recurrent genetic events. And actually, our initial screen, which was prior to this publication of this paper that we'd done just in the first 800 individuals, it turned out that, for just randomly, we had a pretty high concentration of these individuals with 16p11.2 deletions, duplications in that first grouping. It turned out to be about 1% of the whole sample. That became very interesting for us, and we actually started up a whole second study based on that finding, which I'll actually talk about in a moment. So we identified some of these recurrent copy number variations, and that's great. Provide some avenues for us to start thinking about genetically defined subtypes. But there's something else I wanted to do with this set of analyses, which was to sort of look at deletions, duplications, and how that might play out on the phenotype in this autism sample in aggregate. So what I did was looked at, basically aggregated all the duplication cases, all the deletion cases, regardless of the locus of the event. Didn't matter if it was 16p or 1q21. Put all the deletion cases together, put all the duplication cases together, and then look at the relationship between the size of the event, how big is that recurrent event, and how might that relate to IQ and autism severity as measured by a clinical measure we use. And what we found was that the size of the deletion was significantly associated with IQ. So that is, the larger your deletion event, the more impaired you were cognitively. We didn't see that same pattern at all with duplications. Conversely, with our duplication cases, what we found was the larger the size of the event, the greater your autism impairments. That is, the greater your social communication challenges and rate and severity of your repetitive interests and behaviors. But no finding for deletions. So for us, the take home was, well, that's kind of interesting. Not sure what to make of it, other than to think about, well, there seems to be some way, or potential way to think about the role of comorbid intellectual disability, perhaps, and how this is playing a role, as well as just think about potential subtypes that might be defined by the either overexpression of gene proteins, or proteins derived by genes, or underexpression. So something still to explore, but one of the findings from that paper. So while we were doing that, we were still also pursuing some other sort of work to identify genes associated with autism. So we've got our Simon Simplex collection, we're moving forward, we're plugging along, working with all these families. We started up another collaboration that was certainly much more loose than the Simon Simplex collection. That was really just gathering all the folks who'd been working with families with autism, for whom there was DNA available and good phenotypic information. And we all just agreed to pool that together to see if we get the largest sample possible. and then just do a genome-wide association. Just plug everyone into one big pool and say, hey, are there any common hits? Is there anything that's going to identify that there's a potential region, or give us some candidate genes, help us understand the mechanism involved, like just what's going on. So what was exciting about this is it got a lot of attention from Time Magazine when we published this paper, because we worked with over 5,000 families. We did our genome-wide association and found a very strong hit at a small region on the short arm of chromosome 5. In of itself, that's fine. Again, as I mentioned, there's lots of different contributing factors. So for us, it was exciting to at least find something where there seemed to be a relatively common factor. And then we looked closely at what was going on in that region. And there are two particular genes of interest. This particular region was localized right to where two of the cadherin genes are localized. Cadherin is involved in synaptic connectivity and cell adhesion. And so for us, this provided at least a potential pathway or mechanism for what's going on in the brain that's going to then result in autism. So something about synaptic connectivity is happening. That didn't really necessarily help us with thinking about parsing out the heterogeneity, but at least started helping us think about, well, can we make sense of the mechanism? What's the pathway here? So that's moving forward. At the same time, my collaboration with Evan Eichler and his team was growing, and we decided that, based on the versioning technology, that we could actually go ahead and give a shot at doing some exome sequencing. That is, sequencing all the gene coding regions in the genome for some of these families with autism. No one had done this yet before, so we were a little nervous about it given how pricey it was, so we decided we were going to start with 20 families and see what we could find. And so what it is, I selected out 20 individuals for whom it seemed like there was likely that if there were an event, it was a de novo event. It was something that we could make sense of. So I made sure that there was no family history of autism or any mental illness anywhere in the family. I made sure that there was no broader autism phenotype traits in family members. That is, traits that are like autism but don't meet full threshold for autism. I want to make sure there's none of that in siblings or in the parents. And I want to find kids who are significantly impacted with their autism symptomatology. So I identified 20 kids, and then we did full exome sequencing, looked at every gene coding region in these children as well as their parents. And in a sample of 20, we identified 4 genes, significant events where the genes were broken. So gene truncating mutations. And we actually looked at these 4 individuals, and these 4 individuals in this very small sample seem to be significantly more impaired in terms of cognitive abilities, as well as autism severity. So we were excited in that it started, at least this was a potential avenue for making sense of what's happening at the gene level for autism. So given the response in the field to this paper, we actually managed to get some funding to do the same thing in now 210 families with autism. Again, selected out 200 kids following the same procedure who seemed to be significantly impacted, where there's no family history of autism, whose parents didn't seem to have any sub-threshold traits, again, to try to identify de novo or new mutations. And in this sample of 209 individuals, we found 248, almost 250 gene-disrupting mutations, and this occurred in about 25% of the whole sample. So about 50 of the kids had these 250 mutations. So several kids had more than one mutation. That was exciting. We found that, but it was also very sobering because we kind of thought, hey, with 200 families, we're going to nail this. We're going to find lots of things. Because it was the most depressing thing about it was that there was very few recurring mutations. So these 250 mutations, they were all different. And it's like, wow, that's a little bit overwhelming and a little bit depressing. And so we did some calculations based on our sample size, based on the potential genes that we could think about. We came up with an estimate. There's probably somewhere around 800 different autism risk genes that are playing a role. There's another paper that came out in the same journal, the same issue of Nature that this paper came out, and their estimate was about 1,000 different autism risk loci. So a lot of genetic heterogeneity there. But there was something that was at least a glimmer of hope for us. So we found 2 other things in this paper. One was we just found down here in the bottom right, we found that the more mutations that an individual had, the more impaired that individual was. We might sort of just imagine that, but we were able to at least confirm that and demonstrate that. But what was more interesting is we found out that several of the genes that we identified, even though there were very few recurrent genes, many of them were all part of an interactive protein network that involved activity in the beta-catenin pathway. the beta-catenin has a number of roles in the genome, but also plays a role in synaptic connectivity in the way neurons are communicating, connecting. So that provided an avenue for us to explore. So what we decided to do was follow up with a much larger sample, looking at just those genes that we found that were broken, that were operating in this protein-protein interactive network, our beta-catenin pathway as we called it. So we followed up and looked at, in over 2,000 individuals, what's going on with these genes in this beta-catenin pathway. So we tested for disruption of these genes in over 2,000 individuals, and what was at least exciting for us is we started to identify some recurrent mutations now. So with a larger sample, we're actually able to identify, wow, some of these broken genes, we're seeing it in other kiddos. The most common 6 of those, listed here on our figure here on the right, the first 6 genes, when you pull those all together, those account for 1% of all cases of autism. So now we have clearly some recurrence that might be defining at least a small subportion of individuals with autism. 1%, wow, crazy, that's not a lot, but at least it's something. So we were excited about that. So that's kind of that. We just published that earlier this year. So, and we think about kind of where we come since I got started in the 90s in terms of this process. Back when I first got started, we knew the genetic contributors in about 5% of all cases of autism. Those were things like Fragile X, and et cetera, and we could identify that large structural chromosomal abnormalities. In that short amount of time, we've been actually able to increase that to 25%. We now know at least the genetic contribution in a quarter of the kids with autism. Again, it doesn't seem like a lot, but I'm holding on to these baby steps we're making. So, granted, while I was in the Peace Corps, I was a little bit naive, and I was thinking, holy cow, they're going to find all the genes for autism without me. I'm going to miss it. What am I doing here in this hut? And whatever. Clearly, we're still far away. So, but we are making headway. So we've made some headway, but there's still lots further to go. And so what are we doing now then to try to further that and make sense of this heterogeneity and look at these genetically defined subtypes? So I'm going to take us back to a moment to the work with copy number variations. So as I mentioned, we're doing the Simon-Simplex collection. We identified that the most commonly recurring copy number variation is either a duplication or a deletion at chromosome 16p11.2. So we went back to the Simons Foundation and said, hey, we've got something potentially. This looks like it's accounting for about 1% of all cases of autism. That's the most common cause of autism. Let's go explore this. So the foundation got behind us, was very excited, and said, great, we're happy to support you. And we put together the Simons VIP project, that is the Simons Variation in Individuals project. The idea was here, we've got all the different potential contributors to autism, 25% of which we've been able to identify either de novo copy number variations that are specific to a particular family, or family-specific or recurrent gene mutations that I mentioned just a moment ago, as well as some of the other syndromic other findings that we've known about for the past several years. We decided we're going to focus on 16p11.2 and really define and understand that phenotype really carefully from a genotype first approach, as opposed to, again, the way we've done all our genetics work so far is to say, here's an identified group, autism, let's go ahead and figure out what's going on genetically. We decided to say, let's start with 16p11.2, move forward. So we contacted all the genetic counselors across the country, all the medical genetics clinics, and said, hey, there's a network of us. It's our site here at University of Washington, Harvard, and Baylor. We're working together to really understand individuals 16p11.2. Can you tell your patients about us? And really cast a very wide net. We set up a really fancy website so that families could then come immediately find our website, learn about 16p11.2, at least when we started this, kind of what we knew about it, which was very little. And then talk about participation in our research study, which is an in-depth clinical research study. So very quickly, like the Simon-Simplex collection, we're able to work with a large number of families. We just finished our 3rd year of this study, and we've worked with about 200 families, 100 individuals with 16p11.2 deletion, and 100 individuals with 16p11.2 duplication, which just sort of highlights kind of where they are spread out around the country. And the way the study works is, if a family logs into our website, they say, hey, this is our story, we're interested in participating in your research study, we contact them and do a pretty in-depth screening. We collect a blood sample to confirm that in fact the CNV with our breakpoints has been identified, as well as we do cascade testing. We look at everyone else in the family to find out, is this an inherited event, is this a de novo event? Let's find out who has the event in the family. And then we invite everyone in the family to come to one of our 3 sites, and they hang out with us for 3 or 4 days. We take care of all the lodging, we take care of all the flight, we pick them up at the airport, we have packages waiting for them in the hotel, we give them a real nice sort of Seattle welcome, and usually raining, but that's okay. Give them a nice welcome, and then we spend 3 days with them, and we do a very full neurocognitive battery. I'll just go ahead and go to the next slide, or maybe I won't, or maybe it'll just stop. Oh, close. No problem. So what we do is we then do a full battery, a neurocognitive battery that is pretty extensive. And let's see. There we go. Collect real in-depth medical history. Look at the family history. We collect a number of biospecimens, including blood. We also collect do a skin biopsy, so for the development of induced pluripotent stem cells. We collect a number of photographs, including 3D MD imaging, so we can make sure that we're getting looking for any dysmorphology. We do full neurological exam, and we do a full imaging battery, structural and functional imaging, as well as our full diagnostic and neurocognitive battery. What's been really, I think, at least from the family's experience, is that we do all this in 3 days, and then we provide a full feedback with a full report and a lot of recommendations so that families can actually get a little something out of this. For a lot of these families, the experience has been, gosh, we went into the clinic and they told us we had this deletion or duplication, and they said, well, you might end up with autism, or your kiddo might end up with schizophrenia. That's what literature says. Said a little more sensitively, but that's the way I hear it back from the families, and they're reporting to me what they're told. So at least participation in the study allows us to do a full battery that they're not having to pay for at all, and then provide some clear recommendations. And after having us now seen 200 individuals, we actually have a real good understanding of what's going on in this phenotype. So we can actually at least provide some measure of information for the families. So I'll tell you a little about what we found based on this. So this is just a quick table. This is based on the first paper we got just submitted. So looking at deletion cases. So we're just focusing on the individuals with deletions now. 100 individuals with deletions. The majority of them, about 60%, are de novo events. Handful are inherited. But like I said, we collect information on the individual with the event, as well as parents, as well as siblings. So we have full phenotype information on everyone. The idea there is we want to make sure we can understand the familial contribution to the variability we see. So we want to be able to at least somewhat sort of control for that. So what we found in this first 100 individuals with the deletion is that the big take-home is, well, this is the most common cause of autism, at 1% of all cases of autism. Turns out that only about 25% of our sample ended up with a diagnosis of autism. In fact, the most common diagnosis we found was, as we would call it in sort of psychology, phonological processing disorder. But as we look closer, is more realistically apraxia, as we've sort of chatted with a lot of speech folks. As well as second most common finding was developmental coordination disorder, kind of that broad-based sense of motoric difficulties. And then third was language disorders in terms of expressive or receptive language disorders. It was a bit of a surprise for us, but really helped us understand the broad range of the phenotype relative to given genotype. So I'm going to sort of zoom along to highlight a couple of the other findings based on this study. We found that regardless of what type of diagnosis we gave, we found there's certainly a hit in terms of cognitive ability. So our individuals with deletions were the average is about 80, so about a standard deviation below the mean. But if you compare them to their parents, who are doing quite well, these are parents without the event, we had almost 2 standard deviation hit in terms of this cognitive impairment. We see the same sort of finding in terms of social symptomatology, social impairment. So in red here, we got our probands. Again, about almost a 2 standard deviation hit relative to where the siblings are performing on a parent report measure of social ability. So regardless of the diagnosis, we're still seeing a cognitive and social hit with this population. So we've just got preliminary data here for our duplication cases because we're just still doing our data cleaning with that group. But of the clean data, we see a different population. we see far fewer de novo events in our duplications, a lot more inherited events that we're identifying through our cascade testing. So a lot of these parents are pretty typical. We're not seeing anything at all that's atypical about these parents with the same event, and it's these kiddos that are more impaired. Similar to our deletion cases, this is just shown a different way, our kids with the duplications are more impaired than their family members. Again, pretty similarly in terms of cognitive ability. Similar to our deletion cases, again, ASD was not the most common finding for us. Really, again, what we're seeing, in the duplication cases, more predominance of intellectual impairments, but also some the coordination problems and some of the language problems, but not specifically that apraxia, speech sound disorder problem, articulation problem we're seeing in the deletion cases. So that was, again, deletions and duplications. This is a quick summary of some of the differences we found between our duplications and deletions. So in terms of most frequent diagnosis, again, some differences there, with some of the similarities being around impairments in language, receptive and expressive language. Seeing a little bit more of ASD in our deletion cases. IQ is pretty similar. One of the things we found with our people kind of was mirroring what has been reported in the literature in rodents. So there's a paper that came out about a year ago looking at the idea of a mirror phenotype at this particular locus. That is, rodents with a 16p1 point deletion tended to be obese, and rodents with this duplication tended to have a failure to thrive. I don't know if it's still failure to thrive in a rodent, but whatever that sort of corollary would be. So failure to thrive. And we see something similar in that in terms of higher rates of obesity in our deletion cases, as well as greater head size in our deletion cases, where we're seeing much less obesity in our duplication cases and much higher rates of microcephaly in our duplication cases. So a potential maybe mirror phenotype that we're seeing here in people as well. So we didn't want to stop with just 16p1.1.2. We have this massive infrastructure. We have this real neat network that we're able to work together. So we decided we were going to expand to one other CNV, at least in the short term. And we started focusing on individuals with 1q21 deletion or 1q21 duplication. Again, this is one of the other more common causes of autism, accounting for about 0.5% of all cases of autism events at this locus. So this is all very preliminary. We've only seen about 30 or so, just under 30 of these individuals across all 3 sites. I'm just going to highlight real briefly. Here we have just looking at nonverbal IQ and verbal IQ. We've broken, these are all individuals with either deletion or duplication in blue, duplication in green, either children on the left or adults on the right here in each figure. And so no difference between the deletions and duplications in terms of IQ, mirroring what we found in the 16p sample. Certainly kids are more impaired, and this really has to do with, it's sort of a quickly becomes easy to see, really has to do with ascertainment. That is, here on this graph we're looking at IQ again, but now we've spread out across over age, and then who was the initially identified patient. Our initially identified patient, for the most part, were these kids. We identified the parents through cascade testing, and it's only then that did the parents find out, or did we find out that these individuals had this deletion or duplication. So again, suggesting that there's something going on. You can have these events and not end up with any significant impairment. So trying to figure out what's going on with that is kind of our next step. So that's kind of where we're at with the 16p1.2 and 1q21 study. But what we decided to do was actually, because of my interest in trying to parse out and identify genetically defined subtypes, so let's kind of pool all these groups together and see if we can make sense and look at it from an autism perspective. Before I say that, I do want to specify one thing. With our 1q21 sample, I'll just highlight, just like our 16p1.2 sample, a lot of variable in phenotype. But we did see at least a good number, a small number, at least a recurrent event with ASD. And what I'll just say, clinically speaking, just sort of my anecdotal experience is that these kids with 1q21 definitely fit my prototype of what autism was. So these kids walked in and I was like, right, that's autism. When I was seeing all these kids with 16p1.2, I was thinking, gosh, that's maybe autism. It's sort of autism-like. It's a very different feel, just clinically, for these individuals with 1q21 versus the 16p1.2 individuals. So I looked at these genetically defined ASD groups. What I did was I selected out all the individuals from the Simon's VIP study who also had a diagnosis of autism and who had this particular event, as well as I compared it to an age, gender, and IQ matched group of individuals with autism who had no copy number variation, who had no recurrent gene mutation that we could identify. And what this does is it certainly limits the phenotype. Again, as I mentioned, 16p1.2 has a wide phenotype. It's limiting that just to folks with autism. But I want to see if the folks that are coming through to our autism clinics, if we could parse this out in some way genetically. And I looked at all these individuals looking at autism presentation. I looked at phonemic processing. Looked at any psychiatric comorbidities, motor skills, adaptive skills. And well, the take home is it wasn't as clear and silver bullet as I was hoping, but I did find a couple of things. One is that clearly we have this social, kind of a combination of a social hit and a language hit in our individual 16p1.2. So even though to me, having seen hundreds and hundreds and hundreds of ASD kids over the years, they don't fit that prototypical ASD flavor, it makes sense to me that a lot of these kids would end up in an ASD clinic because they have this mild cognitive impairment, a mild social impairment, and then a significant apraxia or dyspraxia or speech sound articulation challenge that kind of makes it difficult for interaction, which then, over time, development and developmental process, sort of pull these kids away from their peers. So it makes sense that a lot of these kids with 16p are going to end up with that ASD diagnosis, or at least look like they have an ASD diagnosis. The 1P2 ones I mentioned definitely had a much more prototypical ASD presentation with that, with impairments of social communication and these repetitive interests and behaviors that we see. And so I was hoping for a smoking bullet with this small little sub-project I was doing, but I'm still hopeful, but I didn't find any smoking bullet there. So I'm doing a number of things that are looking at this currently. So first of all, we've been following up our 16p1.2 deletions, duplications, since any kids that come into study that are under age of 3, we follow longitudinally. And so we've been following them now for about 3 years. We've got about 40 kids that we've been following for several years. And just kind of excited, maybe this time next year I'd actually have some information to share about the longitudinal progression. Also looking more at this apraxia situation with the 16p1.2 dels and dupes. And the other thing we're doing with Simon's VIP is we're expanding more broadly to a much broader online presence to include a number of other CNVs as well. So we can really try to increase our sample size so we can make sense of these genetically defined groups. So in my last minute or so, I'll tell you about, well, last minute or so, I'm going to sort of shut this down for a second, and then I'm going to turn it back on again. What I'm going to do is just kind of pull it all together. We've got, let's see here, right. Essentially what we found in my past 15, 16 years of playing with the genetics of autism, we've identified that there's potentially 1,000 different genetic contributions to autism. It's kind of scary, but maybe we can start poking our way through that. But we really need to examine and identify genetically defined phenotypes because we need to understand how we can best intervene with these folks. We need to be able to find treatments that are going to work for a particular subgroup. What our findings are, those big hits, those big recurrent events like 16p1.2 events, they have wide phenotypic presentation. We have to understand what's going on there. Is that due to secondary hits? Are there other copy number variations, other gene mutations that are acting on that copy number variation that's kind of like a baseline hit on which we build? Or are there other moderator events in terms of common allelic variation that's impacting the expression of those CNVs? So you need to understand that genetic interaction, essentially. And we're going to do that by working with large cohorts and further work with these recurrent gene mutations that we can then see how they're working in an epistatic way with the copy number variations. So that's where we're at. That's a brief overview of kind of my odyssey for the past 15, 16 years in trying to identify genetically defined subtypes. We're not there yet, but we're working on it. So I'm going to leave a few minutes here for any questions that may come up here. Yeah.
So, what proportion, I don't know if you can say this, but what proportion of autism do you think is caused by really de novo mutations versus inherited mutations that are many, many full, of course there's lots of different ones, as you said,
right,
versus a third category, which is de novo mutations on top of inherited mutations?
Right. Yeah, I wish we had some good numbers for that. We're not there yet. I'm hoping actually we can start to answer that at least with these large samples that we can then sort of tease out what this sort of multiple hit is and where is it with inherited mutations versus de novo mutations, how they're interacting. We haven't found the samples yet to be able to answer that, but that's exactly what the kind of information we need to figure out and understand, which will just make it that much more complicated, but that's definitely what we need to be doing. Yeah, there was a question in the back there as well.
You were getting the individuals to the 16
Yeah.
study because they weren't falling into a narrow clinical category.
Right.
How did you get those kids to do that?
Sure. So essentially what we did is we contacted every medical genetics clinic in the country, every genetic counselor in the country. We had lots of postings with any online posting board possible, and just said, if you've been told you have this event, if you have this event, if any of your patients have this event, please send them our way. So it was very widespread. Genetics first. Exactly, yeah, exactly. And there were a couple of families. I mean, one of the families I saw really early on, one of the first families to come in, it was the kiddo was about a year and a half old, and they had found out they had some in utero genetic testing with this infant. There were some questions, potentially there were some potential liver problems and heart problems, so they decided they wanted to do an array in utero. And turns out this kiddo had a 16p11.2 deletion. Also turned out that the findings, the potential worrisome with the liver and other organs actually turned out to be just fine. But the parents were told that when this kiddo was born, well, you know, what the literature says at the time, which was again not very much, there's a great likelihood you'll either end up with autism or schizophrenia. That was kind of the talk of the day. So when this family came in after a year and a half of stressing about their child, what was going to go on to trial, and this kiddo was like, well, at 18 months, super typical. And we've been following him ever since, and this kiddo is progressing totally typically. So that ascertainment plays a huge role in helping us understand what's going on with 16p1.2. Because, gosh, there might be lots more folks out there with this event that we don't even know anything about. Yeah.
And with 1,000 potential targets,
Mm-hmm.
whether it could be different in each case, is there evidence now for multiple vet versus single vet? And is there a way to do sequencing of just those neighborhoods in kids in a way that's more cost effective?
Right, for sure. Yeah, so for sure we know that certainly the presence of double hits seems to be playing a role in increasing sort of the level, the rate, the severity of symptoms. So we know that. We know we've identified potential targets, and we've got this new system that we're using, at least on a research level, with our MIP sequencing we do, where we actually target our specific genes of interest. We can actually do that relatively cheaply. It's not yet ready for prime time clinical presentation, but there are certainly enough events, either copy number variations or genes of interest that we identify, that are part of some of the clinical arrays now. So that's starting to be looked at now to identify multiple hits, single hits, that sort of thing. So it's there. It's not where it needs to be yet, but yeah. It's a great question. So we haven't found that specifically for our 16p1.2 sample. We've found that we see increased paternal age, specifically paternal age, is tied to the rate of these de novo mutations that we're finding at the gene level. So gene truncating events, we're finding higher rates of those. There have been other reports in the literature that increased paternal and maternal age lead to increased rates of de novo copy number variation. We haven't found that yet at all with our 16p1.2 sample, but have found that again with our the genes that we're focusing on specifically in this protein network. But certainly it's kind of an emerging theme that's coming out, is thinking about increased age and specifically paternal age in these genes and regions that are involved in autism.
What do you start thinking about the degree to which the degree that people have babies later in age?
Mm-hmm. Yeah, so there's definitely talk about that, and there was a paper that came out in Iceland earlier this year that looked specifically at, let's look at de novo events, let's look at age, see if we can tie that pattern to increased rates of autism. I think it's a fair thing for us to think about and wonder about. I don't think we've got the definitive smoking gun yet there. I think it's a great theory for us to explore, but just not there yet. Right. Certainly my goal, when I started, my vision was, hey, we're going to be able to identify factors we can screen early. We're going to be able to intervene early now. It's going to be great. I think we'll be able to do that in a portion of individuals, but in many ways at this point, I feel like I'm definitely characterizing the diversity as opposed to making significant headway. However, that being said, that's sort of... well, that's just true. That's true, right? So I shouldn't be such a downer. It's Friday afternoon. I should be fired up. I think we're excited about the notion of these genes that are interacting in this common protein pathway, because then at least we start to think about, okay, if we've got a potential pathway, then we can start to understand that and move forward on a pathway basis, which I think is where we need to go. There's been a couple other pathways that have been identified. Certainly, we've gotten a lot of support for ours in the literature, so we'll keep moving forward that way. That's where, we're hoping for it. Yeah.
Actually, in a more positive way,
Yeah.
we identify that certain loci are associated with these motor and
Mm-hmm.
language problems.
True.
They're actually very amenable to education.
That's a good point.
So,
That's good.
I think it would be very beneficial.
Good. This is great. I like all the support. This is great. We just keep. That's a great point. Yeah.
Very well. Thank you very much.
Yeah. Great. Thank you. Thank you all.